Hello and welcome to this webinar titled Innovation in Biomanufacturing Plants, hosted by Biopharma Asia Magazine and presented by Chris Chen, PhD, CEO at WuXi Biologics, and followed by Dr. Sebastian Revolt, Senior Director of Global Delivery and Sales of BioReliance, End-to-End Solutions at Merck. My name is Stephen Edwards and I'll be your moderator. Before we begin, I would just like to remind our viewers that there will be a live Q&A session at the end of the webinar. Audience members can send their questions in at any time during the webinar via the questions panel below your webinar screen, and we'll go through these at the end of the webinar. Now, please allow me to introduce our first presenter. Dr. Chris Chen is currently Chief Executive Officer at WuXi Biologics, a Hong Kong-listed public company with a market cap of approximately $12 billion. At WuXi, he has built a world-class open-access integrated biologics discovery, development, and manufacturing platform, enabling full spectrum of companies ranging from 2-employee virtual companies to top 20 global large pharma. WuXi has transformed the global biotech industry by enabling entrepreneurs to build successful biotech companies with no need for investing in infrastructure for people, labs, or manufacturing. Under his leadership, WuXi has assembled one of the largest biologics teams with over 4,000 scientists, enabling 60-plus biologics entering into clinical trials and 5-plus products for BLA per year. WuXi Biologics is currently developing 200-plus molecules for global clients. And WuXi has also pioneered disposable bioreactor-based state-of-the-art commercial manufacturing facilities and built the first 30,000-liter bioreactor capacity in the world using disposables. By 2021, more than 220,000 liters bioreactor capacities will be available at WuXi Biologics. I will now be handing over to our first presenter. Welcome, Dr. Chen.
Great, thank you. It's really my pleasure to share the story about WuXi Biologics, how we build biomanufacturing capacity using disposable manufacturing technology and also continuous manufacturing technology. So since the audience this is the outline of my talk. Since the audience are very familiar with the topic, so some of them I will go through them very quickly. Others I'll go into more details. So first, I will still want to introduce disposable bioreactors. We believe it's truly a disruptive technology. And because the scale of disposable bioreactors is very small, so how do you use a small bioreactor for commercial manufacturing? So we have two strategies. One is called scale out. The other one is called continuous processing, and then we'll discuss both. And then we'll share with you about WuXi's vision of what a biotech facility of the future looks like. And then as the moderator just introduced, we actually built a 30,000 liter, the first global commercial manufacturing facility using entirely disposable bioreactor. Total raw volume is 30,000 liter. And we actually built a facility for less than $100 million. We can produce more than 1,000 kilograms of antibody drug substance either for less than $100 million. So hopefully the topic will be interesting to you. I think for now, disposable bioreactor is probably already very much well known. So shown on the upside is really the traditional bioreactors. You see a lot of cleaning, a lot of SIP, and a lot of issues. And showing on the right is a cartoon of current disposable bioreactors. We truly believe disposables are disruptive in that sense because you can save a lot more resource by working on the disposable bioreactor versus a traditional bioreactor. I think this concept is pretty much now widely accepted, so I'm not going to elaborate on this topic more, but I want to share with you how can we use a small disposable bioreactor and to enable commercial manufacturing. So WuXi is one of the pioneers of using disposable bioreactors. Also, right now, we are probably one of the largest users of disposable bioreactors. So back in 2012, we actually built a first facility in the Wuxi city using entirely disposable bioreactors. And this facility was operational in 2014. It only took us 18 months to build a facility. You see the scale. We have 2,000 liter, 1,000 liter fed batch. We also have a provision line. And over the past 6 years, we've been running the facility. we have an amazing 97% cell culture success rate and 96% overall success rate. This is how, if you know how to use disposable bioreactor, you actually can run the facility in a state-of-the-art, very successful fashion. So this is what I call our Wushi's version of a clinical facility of the future. You see, this is the manufacturing facility I just shared with you before, and it only took us 18 months to build. The facility can be entirely disposable, but to show this scalability, we do put a centrifuge in there. So some of the processes use centrifuge, others do not. And this is our version of the clinical facility of the future. The largest scale we can run is 2,000 liter. We can run 2,000 liter fed batch, or we can run 1,000 liter perfusion or 250 liter perfusion. And as I said earlier, that was built back in 2012, and so we've been running it for 6 years. And after 6 years, we have the ton of experience of running facilities using disposable bioreactors. So I want to mention the limitation. Currently, because we're using there is a limit. Currently, the largest size probably 4,000 liter. That may be already the maximum you can do with a disposable bioreactor. And because it's a disposable, so you actually do manual installation. So training is actually very critical. So we have been running 97% success rate. Training is crucial part of the success. And if you have a process, if you have a new cell line that has not been exposed to the disposable bioreactor, you may have some issue with the extractable leachables. But for Wuxi, because everything is based on disposable, so we actually screen cell lines. That's part of our cell line development process. We actually screen cell lines that perform well in disposable bioreactor. So that's not an issue for us. And today we have Merck here. I think we have been using a disposable bioreactor from all vendors, and then some of the vendors, suppliers still need to improve on robustness of the supply chain. So as I mentioned earlier, the topic of today's discussion is how do we use disposable bioreactor for commercial manufacturing. So because each unit volume is small, it's in 2,000 liter, 4,000 liter, and then how can you use that for commercial manufacturing? We have laid out two strategies. One is called scale out. The other one is continuous manufacturing. So I'll elaborate on each one of them. The concept of scale out is actually pretty straightforward. So when you have a program in phase 1 and you use one reactor, once you get to phase 3 or commercial, you use up to 6. So with a 2,000 liter bioreactor, now you can actually run a batch of 12,000 liter. So if you have a 4,000 liter, you can actually run a disposable bioreactor with a batch size of 24,000 liter. So it's comparable to a traditional stainless steel, whether it's 12,000 liter or 24,000 liter. And that's the concept of scale out. The good thing about this is, essentially your product quality is mostly determined on upstream in the bioreactor. If your phase 1 and phase 3 and commercial is the same bioreactor, so your risk of comparability issue is totally eliminated. Basically, your phase 1 and phase 3 is the same bioreactor, same cell culture process, and that will eliminate a lot of issues down the road with the scale up. So why do we need to scale out? And so as I mentioned earlier, so scale out gives you a lot of flexibility. So in phase 1, you can run one reactor. In phase 3, you can choose 1, you can choose 3, you can choose 6. And that gives you essentially from 2,000 liter scale all the way to 12,000 liter scale when you're in phase 3. Again, the product quality, there's no change. It's the same product quality. And because of that, you can actually adapt to rapidly changing and unpredictable market demand. You can also build a much smaller facility because using disposable is smaller and modular facility. You can actually put them in different regions. You can put one facility in the US, one facility in China, one facility in Europe, and that's Wushi's strategy. Our global network, we can actually copy paste a facility from China, put it in Ireland, or put it in US. So as I hinted earlier, because you don't need to scale up, you actually don't have any issue associated with the PCO2, with the dissolved oxygen, with the mixing. The product quality of your phase 1 material is identical to your product quality at phase 3 and commercial. So this makes your product launch so much easier and so much more straightforward. So I already mentioned the benefit of scale out. I think that it's a stepping stone from phase 1 to phase 3 and eliminate the risk of cell culture scale up, no comparability issue. You can grow the demand as your product volume needs. And you can also, because you're using disposable, you can have more batches from the facility. And again, the same facility can actually run multiple scale. So one thing I didn't highlight is actually the same facility can handle a very old process of 0.5 gram per liter or 1 gram per liter, or very state-of-the-art process with 10 gram per liter. Traditionally, if you have a stainless steel bioreactor-based facility, it's very hard to run 0.5 gram this batch and then 10 gram next batch. With disposable, you can. Because for 0.5 gram, you run them at 12,000 liter scale. For 10 gram, you run them at 2,000 liter scale. That gives you a lot more flexibility in running a facility. For CMOs like Wuxi, especially, it's very beneficial for that. So the advantage of scale out, again, is parallel production lines, standardized facilities, smaller footprint, modular architecture. You can tech transfer very easily. Again, because it's the same bioreactor handling your phase 1 and phase 3 and commercial. You can put in multiple facilities globally. That's what we believe the scale out will give you the advantage of that. So scale out address the issue. How can you use multiple bioreactor to produce commercial product? Another way to do this actually is implementing continuous processing. I think all of us have been very familiar with FDA trying to drive continuous processing. I think in small molecule drugs, there are already quite a few approved products already using continuous processing. But for us in the biologics field, continuous processing is actually not that rare. So a lot of the old product, actually the legacy product, was actually made using continuous processing. But that was by necessity. Otherwise, the process wouldn't work. So now actually continuous processing is probably can with the disposable technology, continuous processing give you a new insight. And then you use the same 2,000 liter bioreactor, you can actually produce the same amount of product as a stainless steel of 20,000 liter scale. And that's why continuous processing is coming back. It's also getting very popular. So for lower volume, if you have a product with a 205 kilogram of antibody demand, you can actually use 1 or 2 bioreactors. You don't need a huge facility. And because of the continuous processing, your cost is lower, and I will explain to you later on why the cost is lower. Yeah. So this is a traditional continuous processing. Again, as I said earlier, so perfusion has been used in the industry for 30 years. So at Wuxi, we not only do continuous processing on upstream, we actually continuous to capture. So we incorporate the protein A also is continuous. This way, the whole unit operation is not being integrated. So every day you perfuse and you actually capture them in the first column. This way, your efficiency is much higher than a traditional batch processing. This is some of the examples that we have achieved. With the continuous processing, we can actually increase the cell density to 100 million per mil. And that actually results in every day we get 3 grams per liter per day. Every day we get 3 grams per liter. So as we know, traditional fed batch, for most of the process, after 14 days, you get 3 grams per liter. So in a way, this technology gives you 10 times more productivity with the same volume of a bioreactor. And that's why we believe this is potentially a way of the future. This is shown here is a few examples, and we actually run them in labs, and we also recently scaled them up into a GMP setting. So you can see in this case, we are running 30 cycles for protein A. We've seen very consistent behavior. Later, we actually run it all the way to 100 cycles and showing very similar data. And this is the continuous capture, and you see the protein A column, the continuous capture site. You see very consistent yield, consistent quality for the perfusion. So with continuous processing, you can actually control product quality a lot better than the traditional fed batch, because fed batch, what you get is a mixture of day 1 product versus day 14 product. For perfusion, if you run it in a steady state, every day the product quality is fairly similar. So that's the 2 approaches Wuxi want to use to essentially scale up our using disposable bioreactors for commercial manufacturing. So about 4 years ago, we said, Wuxi is focused on disposable. How do we define a facility in 2020? How does the facility look like? So we put all our requests in or maybe a demand on facility of the future. How does it look like? You need to be very flexible, can handle fed batch, concentrated fed batch or perfusion, can handle both grandfather in low titer process or recently high titer process, and can need to be state of the art and need to be leverage the best of disposable and stainless steel, and need to be highly automated. Need to have eventually MES and then all the industry 4.0 put in there, and certainly it's modular so that you can actually put in different locations. one in US, one in Europe, one in Asia. And lastly, you have to become cost competitive. So I gave my team an assignment saying, can you design a facility to produce 1 metric ton of antibody for less than 100 million? And that's the assignment that's the sort of challenge we took on about 4 years ago. And here today I report the results that we have. So that's essentially become the result of our facility number 2. It's called MFG2. It's a multi-product facility, can run different process and can run process with different productivity. And we use both scale out strategy and also continuous processing technology. So in the scale out, we actually have 14 2,000-liter bioreactor in there. We have 2 trains of each train with 7 2,000-liter bioreactor. So we can run the process as 2,000-liter scale, 6,000-liter scale, or 12,000-liter scale. And then we have 2 by 1,000-liter perfusion. So each suite can actually produce about 1 metric ton of antibody per year. And the capex for each one of them is actually less than $100 million. So the perfusion facility, it cost us $65 million to build. The fed batch cost us $85 million. Both facilities are already running. So I'm going to share with you some of the exciting pictures from the facility. So this is the facility set up. So we as I mentioned earlier, there's actually 2 facilities. So we on the upstream, you see different technology. Downstream is very similar. Downstream, we actually use traditional stainless steel. Upstream is entirely disposable. So on the facility A is the fed batch facility. On facility B is the perfusion facility. For perfusion facility, we can also run them in traditional fed batch mode as well. So we started this in about 2014, and we put the design into practice. We actually have a campus with about almost 500,000 square foot facility, and we have it's 500,000 square foot of facility. You see in the middle is actually the main facility. We have the warehouse, and then the GMP cell banking area, and the utilities all in this one big campus, and certainly the QA and the QC is in there as well. So we made an announcement. We said we have the ambition to build, at that time, the largest mammalian cell culture manufacturing facility using disposable bioreactors. The total volume is 30,000 liters. And then the total capital is $150 million for this project. And this is the same setup as you see before. And this is actually the picture of the entire site, about 500,000 square foot facility. On the smaller picture, you see actually this is the picture inside of the facility. You'll see a lot more in next couple of minutes. So this is actually our what I call a version 1.0 commercial facility. So the perfusion, one upstream and 1 downstream. Fed batch, 2 upstream, 1 downstream. So fed batch, you can run them at 2,000 liter scale, 6,000 liter scale, or 14,000 liter scale. So it's globally GMP compliant. It can handle using hybrid technology, and it's using traditional room classification. The process is actually highly automated. We have a Delta V system, and then on top of that, we have a MSI, OSI Pi historian to capture all the data, and then the facility can eventually incorporate into MES. So this is a layout of the first floor. The perfusion and fed batch are almost side by side. On the first floor, you see all the bioreactors in there. And the second floor is actually mostly the seed train. First floor is the bioreactor and purification. Second floor is the seed train. And as I mentioned earlier, the facility is already highly automated, and then we can actually run MES eventually. This is a very high-level timeline. It only took us about less than 2 years to build. We started this. We had a concept in 2014, and then we started conceptual design in Q3 of 2014, and we actually run the facility in 2016. So this is the upstream, entirely disposable. You see the bioreactors in there, and you see. The purification column is actually very large. It's a 1 meter column, and so we do it semi-continuously. Now we also implemented the PCC, the continuous flow systems in the facility. And then, as I said, the facility also leverage a lot more stainless steel because of the buffer volume, media volume. So media and buffer is actually traditional stainless steel. You see big tanks, 5,000 liter tanks, stainless steel in there. And the facility you see in April of 2016, actually you see you can still see the flower from the, and then you see all the construction pictures in there. You see room classification changes. This is the second part of the facility. So the first part was built in 2014. The second part was built in 2016. The fed batch piece. You see, as I said, media and buffer is a really super scaled of a stainless steel system. You see them in there. And so that's actually our version 1.0 process. So we run the perfusion process for 4 years already. We run the fed batch process for 2 years. So now we actually took on the effort saying, can we go even more bolder? Can we go even bigger? So we actually scaled up this again to Ireland. We put a much 2x, almost twice the size of the facility in China. We put it in Ireland. So in this case, now we're going with a 4,000 liter disposable bioreactor for fed batch. We're still going with a 1,000 liter for perfusion. So it's almost twice the size of what we have in what I have shown you in China. And this is what I'll call a version 2.0 process, and it's larger, and it's also more automated, and it's also all the lessons learned we have from the first facility hopefully will be captured in the second facility. You see, this is the first level. You actually see the 2 suites of each one of them, 3 or 1,000 liter bioreactor. With the WuXi's continuous processing technology, you can actually this suite can actually produce 3,000 kilogram of antibody. So that's a lot of antibody. 3,000 kilogram probably correspond to $3 to $5 billion sales of a product. And then the fed batch is actually 4,000 liter, and so again, give you the flexibility. We can run as small as 8,000 liter scale, as big as 24,000 liter scale. And then the cost come from those facilities actually is very comparable to any global companies. Again, the companies with 1 to 5, 15 to 20,000 liter bioreactors, 1 to 6. Facility you see, this is a cross-section view. You see still a lot of the stainless steel tanks because of the volume, the number of buffer and volume media you need to process. So we are looking at technologies probably like inline dilution technology to hopefully reduce some of the buffer and volume in the future. But currently, this is still very traditional technology. And you see this is our site in Ireland. We just started to build. And end of next year, this will actually be a fully functional and we're ready for GMP validation facility. So right now, we purchased 5,000 tons of steel using for this site. We're on track to have a GMP production Q2 of 2021. We'll be ready for engineering rounds end of next year. I think looking into the future, I think with the disposable technology, with the continuous processing technology, I think we believe the titer can achieve 40 to 50 grams per liter. We have already done that in the lab. In the lab, we have already achieved 50 grams per liter using the 1,000 liter, using the perfusion technology. Essentially, every day we get more than 3 grams per liter. And then with this technology, we believe we can reduce the cost of antibody, drug substance from traditional $200 to $50 per gram, maybe even $15 per gram eventually. On the right side, you can actually see two charts. This is actually the cost of penicillin over the past 30 years. You see a huge reduction of cost of penicillin from a couple $1,000 per gram to actually a couple $1,000 per kilogram or even per metric ton eventually. So I think hopefully biology will also be able to achieve huge cost of goods reduction with those technology, and that eventually will benefit patients who need the drug most. Yeah, so that's basically WuXi's vision, how we believe the future facility will look like, and how our version 1.0 process or version 2.0 process will look like. I want to use a few minutes to basically share with you about Wushi Biologics if you're not familiar with us. We are a global CDMO. We have a very large team. Now this year we have close to 5,000 people, and we have about 2,000 people working on IND enabling work. So currently we have more than 200 customers from globally, including 13 of the top 20 large pharma. Companies like Genentech, Eli Lilly, and Celgene, Johnson & Johnson, AstraZeneca, both work with us and all work with us in the public domain. So we actually have a capacity to enable 60 INDs per year, 3 to 5 BLAs per year. So last year we help our clients filed 45 INDs. So this one company help our client file 45 INDs. So currently we already have 200 molecules in development. So we are helping global companies developing 200 molecules, and we have been audited by global clients. We are the only FDA and EMA approved facilities in China. So as I said earlier, we have a huge pipeline. So we're helping global companies developing 205 molecules. Among the 205 molecules, about 70% actually come from US and European companies. So we have 97 programs in preclinical stage, 61 in phase 1, 26 in phase 2, and now 13 in phase 3, and we have 1 commercial product. So last year alone, we actually added 57 molecules. So we have a huge market share globally. And as we also have 10 molecules transferred from competitors, including some of the phase 3 programs. So I want to summarize what I talk, and so I think I truly believe, and Wuxi is totally counting on disposable bioreactors. We truly believe it's a disruptive technology. We believe significant advantages and in the disposable bioreactors, and also the new investment in continuous processing can really enable integrated bioprocessing for proof of concept commercial facilities. Both scale out and continuous manufacturing will help really reduce cost of goods for biologics manufacturing, eventually will benefit the patient. I think what Wuxi has been a pioneer in this area. Will continue to invest, and we will be able to make large, high volume antibodies, biosimilars, low volume and programs, enzyme replacement therapies, you name it. I believe this technology will really help transform the industry. And with that, I want to acknowledge my commercial facility project team and my continuous processing team. I will thank you all for your attention.
Thank you, Dr. Chen, for your presentation on innovation in biomanufacturing plants. Now, before I introduce our second speaker, I would like to remind our audience that there is still a question and answer session at the end of the webinar. Please feel free to send in your questions at any time during the webinar via the questions tab located directly below your webinar screen. And we'll go through these at the end of the presentation. Now, please allow me to introduce our second speaker. Dr. Sebastian Revolt leads activities including the biodevelopment, including development and production of clinical API for internal and external clients. Sebastian manages the operations team as well as quality, regulatory, analytics, and the associated support groups. He also supports the development of new and innovative biomanufacturing technologies and solutions through new technology activities of the process development team. Dr. Ribault received his microbiology engineer degree in 1997 from the Engineer School of Luminy, Marseille, France, and his PhD in molecular and cellular biology in 2001 from the University of Strasbourg. I will now be handing over to our second presenter. Welcome, Dr. Ribault.
Thank you, Stefan, for the nice introduction, and thanks to the audience for being on that webinar today. Very happy to present you what activities we're running using single-use technologies, including some consideration on continuous map processing, but also on what the manufacturing of the future could be in terms of closed processes. So maybe I can start with briefly introducing our organization, our experience and expertise. We are a contract development and manufacturing organization that was created in 1987, so about 30 years ago, with a clear focus on process development, but we also received our first GMP certificate 24 years ago in 1995. We have developed over 250 biologics, some of them being commercial products today. If I look at the number of batches we have run over the past 7 years, it's about 70 plus unique GMP drug substance batches released since 2012. So what unique means we had repeats for some molecules, obviously, but I just want to show that we run about 70 different molecules, which means 70 different experiences running the processes in GMP using single-use technology. We have an engineering branch in our activity so that we can develop quickly our facility, but also provide that services to our clients. So we typically build a new single-use facility in less than a year. One concrete example today, we're going to break ground for a new GMP facility for our organization in Shanghai, in China. We'll break ground next month, and the facility will be up and running and delivering the first GMP batch by the second quarter next year. In terms of technology expertise, we're CDMO as well as a developer and manufacturer of process technologies. I think that most of the audience knows that Merck is working a lot on single-use technologies. We have worked historically on 90% original molecule, 10% biosimilar, and we enjoy 100% scale-up success rate. We scale up directly from a 3-liter bench scale to 2,000-liter GMP manufacturing scale, and our processes are about for 2/3 fed batch and 1/3 perfusion. We are based globally, but we deliver locally. So we have a process development and pilot manufacturing site close to Boston in the US. Our main facility is in Bordeaux in France, where we conduct activity from cell line development to GMP manufacturing. Our GMP scale is multiples of 2,000 liter. We can provide a 2,000-liter batch as we can provide a multiple of 2,000 liter. Since we developed back in 2012 the strategy to run in parallel multiple large-scale runs, I have in mind an experience of running parallel thousands of liters batches, 36 times for molecule that is marketed now. And finally, our last location is Shanghai in China, where today we execute from cell line development to pilot manufacturing, and where, as I mentioned before, we're building a facility which will be multiples of 2,000 liters. So if you look at the history of the implementation of stainless steel and single use in our organization, I think it's important to mention that we started with stainless steel and that we transitioned to single use. Why is that important? Because we accumulate these two experiences. We know how painful it can be to run a stainless steel facility with very little flexibility, and we really measured the benefit of having a single use implementation. So our site originally, and you can see that on the right side of the slide, was running upstream at 200-liter stainless steel. We upgraded the facility in 2007, running a bioreactor at 1,250 liter twice, and again stainless steel. And starting in 2011, we started the revamping of the facility to introduce first 200 liter, and then we were running a hybrid facility. By 2015, we completely eliminated stainless steel and moved to 2,000 liter single use using the Merck Modus bioreactors. In 2017, we expanded the facility to implement again 2,000 liter suites, and the 2,000 liter suites are designed to host two 2,000 liters so that we can run 4,000 liter batches, and obviously we have the DSP aligned with that. I won't enter into a lot of details around the facility. It's something that I will be able to discuss offline. But briefly, the facility is split between clinical runs on one side and commercial runs on the other. Talking about the benefits of moving to single use, I tried to illustrate that on that one slide, where you see at the top of the slide the typical time for a suite at 1,250 liter stainless steel, and at the bottom of the slide, you see a suite at 2,000 liter run. So there was a lot of time working with stainless steel, preparing the bioreactor, setting up the suite, conditioning the media, cleaning, sterilizing, and so on. And that turnaround time is gone with single use. So we observed that we could increase the manufacturing capacity by about 50%, moving to single use, using less operators, having training much more quick and efficient. And we've actually designed a specialized training for single-use equipment, something that we do internally, but we also train a lot of our customers who want to tech transfer in the process in the end, and we train their operator in GMP conditions, specifically with single use. I mentioned earlier that we have aligned our platform and we're scaling it directly from 3 to 2,000 liter, and the graph at the top right of the slide is showing the alignment between all our platforms, the 3 liter, the 200 liter, and the 2,000 liter. Benefits of single use, obviously, increasing the capacity per year with the elimination of what we call the non-added value time, also a cost saving per batch. That we have modelized first using softwares like BioSolve. But not only we modelized before the change, but we have measured after the change, and we've seen that the criteria we entered into the model, the data we entered into the model, translate into the real life in terms of savings. So we save on electricity, we save on water, we save on a smaller number of technicians needed to apply the process. We save on no spare parts anymore for stainless steel, and just having our single-use inventory, which is quite easy to manage. It's important when you change from stainless steel to single-use, or when within single-use you change from classical manufacturing to perfusion manufacturing or continuous manufacturing, to look first at what is the maximum production scale you need, really. Don't overscale or don't underscale. Work on your supply management and waste management, because they are going to change a lot. So we spend a lot of time working on how we would implement the equipment, really designed, as I said, the training around handling, but we've seen that handling was reduced. There's no cross-contamination since we're working in an environment that is pretty much closed, and we've seen that we were extremely reactive, and when someone was asking us for a new run, we were able to restart activities very, very quickly. So single-use systems are really fit for flexible activities and high productivity system. Again, we've modelized it and we observe it now since 2012 when we moved to single use. When we speak with in general, scientists, developers, and manufacturers, we observe the following drivers for the adoption of next-generation processing technologies. Flexibility comes as a number one. 93% of the people want more flexibility. 90% want to control the cost and reduce the COGS. 77 want to work on a pipeline where multiple molecules exist. And finally, 73% want to work in a facility, what we call multi-use product facility, and I will cover that in the last part of that presentation. So what is next generation processing? There are many definitions. We came up with one. Next generation processing is the use of any technology, expandable service or system, which significantly changes the existing monoclonal antibody manufacturing templates to deliver more speed, more flexibility, an increase in quality, and a decrease in cost. We clearly see an evolution that we can predict. The facility goals are less footprint, about 80% less, and it's something we've seen when we moved here in Europe from stainless steel to single use, and it's something we experience as well today, building our facility in Shanghai. If we had designed the same facility 20 years ago, the footprint would be massively more important. We also reduce the capital. We see a decrease that can be up to 75%. Construction time is down by a lot. I said below a year for the construction of a facility, when we were used to build facility in 24 to 36 months, and a reduction of OPEX by about 60%. What are the enablers? The intensified processing, single-use technology, process analytical technologies and software and control use of technology for inline, at-line monitoring, for example. That changed the game quite a lot. At the bottom of the slide, you see the timeline, process, format, controls, analytics, and software today, near-term, mid-term, and end state. I won't elaborate on all the briefs, but if we look at what we're doing today, we run a batch. Many people still use stainless steel. We have process monitoring strategies in place. Analytics is run by QC, and software is more or less by unit operation. What's the end state? Continuous processing, closed processing, at least connected technologies in a format that is single-use and primarily closed. Controls are based on holistic process control. Analytics are inline, real-time, at-line, and we use softwares for process simulation. If you look at the train today in 2018, 19, and where it evolves going up to 2020, we are reducing the number of operations. Less bioreactors running in perfusion, having continuous chromatography, inline virus inactivation and flow-through polishing. And the end state should be probably after 2025, what we could call the magic box, where you enter the cells on one side and finally you get a fully purified drug substance in the end. So focus area are listed here. Cell culture media for intensified process, integrated perfusion bioreactors, the continuous purification using protein F for intensified capture. Buffer management is an important topic as well as flow-through polishing and single-pass UFDF. What are the upstream intensification strategies? I won't go through the entire train, upstream and downstream, but I want to focus on a couple of areas. So let's start with upstream. There are two key areas. Intensified seed is number one. Working with high cell density process intermediates and a perfused seed train. Having more cells very early in the process so that we can feed very quickly the end bioreactor for production. Which brings me to the second bucket, that is intensified production with a high seed fed batch or concentrated fed batch, ultra high VCD fed batch, or steady state perfusion and dynamic perfusion. That's how we're moving from the processes that we've all seen 10 years ago, 0.5 gram to 1 gram per liter, moving now to the current processes where our cell line platform can achieve 5 gram per liter, 8 gram per liter, and moving to steady state perfusion with 20, 30, 40 grams per liter. So when we look at these intensified seed train or perfused seed train, how is that going to translate? You have an illustration here. We're going to reduce the high seed fed batch, for example, moving to 14 days to 10 days. I know it doesn't look like a lot, but we're talking of about 30% time reduction, which means the ability to run a significantly higher number of batches every year. So we'll implement cell retention devices so that we can concentrate the cells in a compressed seed train and decrease the overall time. So what compressed seed train means? Means facility utilization is reduced, higher number of batches, footprint is reduced as well, which means cost of operating will be smaller as well. High seed means increased manufacturing capacity and increased facility utilization as well. Obviously, we model these new upstream conditions, and we enter a number of parameters that are illustrated here. We usually modelize using the BioSolve softwares, and what we see from this modelization is that indeed will increase the manufacturing capacity and the facility utilization. If you go and modelize with all the assumptions that are listed here, that a conventional train with an N-2, -1 production and harvest will bring you up to 5 grams per liter, and that with a high seed train we're going to use would be 4 times smaller, 50 instead of 200. The N-1 would be smaller, 200 instead of 500. production batch stays the same, but we have 10 days instead of 14 days. So we entered all of that into our modeling software, changed the volumes, changed the C-train scenario, and proceeded with all of that. This is what we see. We see a clear reduction of capital due to smaller vessels for C-train. We see an increased operational cost due to higher media requirement, but overall, that translates already now, and I'm not talking about the fully continuous process, I'm talking about things we can do now. That translates into a reduction of COGS that is slightly below 12% with the high C-train scenario. And all of that is indicated on the slide. So you see the capital reduction, you see the difference in material, consumable, labor, and other. I won't discuss all these numbers in details, and we'll stay on the take-home message that is just below 12% COGS reduction. Many times, I have the question, okay, moving to that, there's more media, there's more buffers, there's more physics, etc. Don't you think it's going to be more expensive? The answer is no, and we see that reduction here. Moving to the impact on the plant throughput, you see the orange curve, that is the high seed configuration versus the blue curve, that is the current fed batch scenario. So we see that the high seed train scenario provides a shorter time in production bioreactor, which means increased batches per year, and we can measure it here. As you see, the blue curve in batches per year. When using the high seed train, we're over 120, we're at 125. So keep these two numbers in mind. 90 batches per year in a typical batch setup versus 125. So we're talking about a 30% increased manufacturing capacity, reduction in COGS, increased capacity. Let's move now to downstream processes. Again, many unit operations. I won't mention everything. Let's start with capture. You see here one of our new equipment currently being developed by the R&D team within Merck. Continuous multi-column chromatography, impurity clearance, and obviously performance versus cycle numbers. And the system we developed is fully disposable flow path. 3-column operation with about 30 days of continuous operations. We're talking 8 to 120 liters per hour per line with 3 product inlets and 6 buffer inlets. We obviously have flow BP detectors on the outlets. So you see at the top of the slide the system as it will be when fully finished, and you see today the system that exists. As you see, we're really close from the final configuration. It's a very interesting ongoing development. Another unit operation that you see here is virus inactivation. Today in a batch mode, the industry standard that I'm sure you all know, 60 minutes hold, robust inactivation with above 4 LRV, 2 holding tanks required, and very manual process. When we move to continuous, we're talking about continuous pH adjustment, dynamic hold. This eliminates large holding tanks. It's automated and enables continuous operations. The applicability is dependent upon the virus inactivation kinetic, obviously, but also the efficient incubation chamber design. We're part of a large project called Horizon 2020, funded by Europe. It's a program for research and innovation, and here's the final process recommendation. So I will focus on the bottom part of the slide. That is the new process. We're talking of a bioreactor with connected and continuous processing, moving to harvest, continuous capture, then going to continuous viral inactivation, moving into polishing after, and finally UFDF. Fully single-use unit operation, innovative technology. We clearly see on that graph the reduction of time, since the scale of that line really represents the time savings. So we're going to move from disconnected to connected, from some activities that are still open for many of them to activities that will be closed in the future. We already have a proof of concept, and you see it here. I'm sure that you will recognize on the left the skid that we've seen before for continuous capture. We have the skid for viral inactivation that you see in the middle, then polishing, and the only thing that is not on the slide here is the UFDF. So as you see, proof of concept exists. So already aligned with a 1,000 liter scale setup. So why not 2,000 liter? Because it's a proof of concept, but obviously scaling from 1,000 to 2,000 is not going to be the difficulty. The difficulty was to come up with these systems. And as you see them here, I mean, they are real. We're not talking about 3D modeling. These are real systems in a real manufacturing environment. So what are the advantages of that program? Capability of processing 3,000 liter harvest in 24 hours. So we're talking about 12 kilograms of mAbs in 24 hours with a footprint that is really reduced 10 to 15 times smaller than what we have today in DSP. Elimination of large intermediate oil tanks is one of the reasons of that decrease in footprint. Flexibility, much more important with mobile equipment. Single use means reduction of bioburden risk, no carryover issues, no cleaning, regeneration, steaming, and rapid changeover. In terms of economic, we're talking about 35% less in the facility investment, 30% less running cost, and cost of material decreasing by about 50%. In terms of environment, reduction of the CO2 emission by about 25% and reduction of water buffer and resin consumption with a decrease of water by a factor of 10. That is massive, and that was very important for us to be able to reduce this environmental impact. Final part of this presentation, we're talking about continuous, we're talking about closed. What about multi-product? So, I mean, what is multi-product manufacturing first? It's the ability to manufacture different products in the same room and at the same time. So we're working on this concept. We don't want to stop our thought process to just continuous manufacturing, but want to have a look also at would we be able in the future, near future or long term, to run multiple products in the same room. Today, upstream and downstream are fully separated, as you know, and even downstream pre and post virus are separate. The ability to run different products in the same suite means more flexibility, time saving, and cost saving. So what's the purpose of that project? It's to see if we could run in parallel in upstream and in downstream 3 different products, as it's illustrated here on this slide. The 3 products are running in the same upstream suites, and the 3 same products would be running in parallel in the same downstream suite. What does that mean? If you look today at this schematic, it illustrates the way a typical facility runs a biotech process. Upstream is grade D, downstream is grade C. We have pressure cascade. We have some environments that are considered as contaminated, like washing area or in-process control, for example. So moving to a process where we would have multiple products in the same suites means lots of changes. What are the GMP requirements today? We need to move from one suite to another, respecting the cascade pressure. A sample that is going from upstream to in-process control never goes back, because in-process control is seen as a contaminated environment. If we move to multiple products, the production area of tomorrow would be very, very different. Actually, the contaminated environment would stay about the same with the washroom and in-process control as well. But we could consider, since the process would be closed, that the upstream and downstream suites would be seen as CNT, as clean and not a controlled area. So that difference between grade C and grade D disappears completely when we talk about upstream and downstream, which is an interesting thought. It's something that is ongoing. We're looking also at the concept of ballrooms, where upstream and downstream are in the same suite. The more we'll move to continuous manufacturing, the more we'll move to closed processes, the more we'll be able to think about these fully closed multi-product facilities. I will finish with that summary slide. It's a journey. to shape the future of bioprocessing with improvements to unit operation, with intensification of these units. We're talking about fully connected and continuous processing. Today, we're talking about contiguous processing already, with unit operations being connected one with the following. The perfusion operation in seed train and production bioreactor can shorten the overall process time as we've seen it and increase the product titer. They also enable the continuous processing with constant harvest. And through the use of the process modeling that I mentioned several times in this presentation, we see significant improvements. COGS reduction, I mentioned that. Today, about 5 to 10%. We know we can do better. Footprint reduction that is really significant through the reduced buffer volume and whole tanks, and about 72% reduction in processing time with flow-through polishing. I'm going to stop here, thanking again the audience for listening, and obviously open to any questions. Thank you all.
Thank you, Dr. Ribault. As you mentioned, now we will begin the question and answer segment of the webinar. But once again, I would like to remind the audience that you can still send your questions in via the questions tab located directly below your webinar screen. So now our first question. Would Disposable be able to compete with 15K or 20K stainless bioreactor in terms of COGs? And that question is for Dr. Chen.
Yeah, as I mentioned in the seminar, that absolutely, with the scale out and with the continuous processing, so we can actually compete with any current fully established facility. So that's the beauty of as the second speaker also mentioned. So with the disposable, your capex is lower, your facility is more output, more throughput, but in the meantime, your cost of goods actually is still lower, comparable or lower. That's why we believe disposable is truly disruptive technology.
Thank you. Our next question is actually, what are the challenges? I believe that's a follow-up question for you, Dr. Chen.
The challenges for building disposable facilities is actually mostly because of the manual operation. It's not prone to high degree of automation. That's why we're trying to automate as much as we can. So the training of the people is absolutely critical. And the second one is actually how robust your supply is. With a stainless steel bioreactor, it's fixed. Once you purchase the hardware, the reactor is fixed. With a disposable bioreactor, every batch you receive from your vendor may be slightly different. So how do you ensure supply? How does our vendors or our partners ensure that their disposable bioreactor they produce is batch to batch is very consistent? So that's actually the two main challenges.
Thank you. Our next question is, where do you see continuous bioprocessing being the most/less used? I believe that's for you, Dr. Revolt.
It's a good question. Most use on products where a very large quantity is needed. So we're talking about, we've seen a couple of projects where the needs will be at the peak at the metric ton levels. We're not talking kilos, we're really talking tons of products per year. So in that case, it requires in the old systems the 15 to 25,000 liter bioreactor. When using continuous processing, we would be able to do that with what we call the 4-packs suites, where we would have 4 times 2,000 liter continuous processes running, and that would be able to achieve the metric ton. So that's where I see that it's the most useful. The less useful, more and more we have projects touching what tends to be personalized medicine, or at least niche indications, with about 1,000 to 10,000 patients treated per year. And that's a need of a kilo to 10 kilo per year. In that case, continuous manufacturing doesn't really make sense, as a 2,000 liter bioreactor, one batch is providing you with the necessary quantity. I must say that although I'm saying that continuous manufacturing would be less useful in that case, I'm not saying it wouldn't be useful, because if we want to be more flexible and agile, still it would make sense to run these processes in a continuous manner so that we can run in our facility more and more batches and reply to the increasing demand. So again, I'm not saying not useful, I'm saying less useful than when you need huge quantities.
Thank you. Our next question is, can you tell us more about advantages of closed processes that you enjoy today? Again, that's for you, Dr. Revolt.
First advantage, closed means no door for any contamination. So it's obviously one of the huge benefits. I remember when we were running stainless steel, how many points we had to check before we were starting, how many points we had to clean in place, and so on. When using closed and single-use system, you have an irradiated, pre-assembled system that comes and it's ready to run, so risk of contamination decreases a lot. Other advantage, since it's closed, pre-assembled, less training for the operators, less chances of making a mistake, since everything is ready, plug and play. Last advantage I see is that whenever we have discussions with some of our clients who are starting in the biotech activity, it's very easy to understand that overall a closed system is safer and easier to manipulate. So it's not only our point of view as a CDMO and as a CMO, it's also the view of our clients. We can see the benefits of having these systems versus the historical stainless steel part.
Thank you. Moving on to our next question. With growing manufacture capacity, buffer volumes are bigger and bigger. How does WuXi overcome the large volumes of buffer? I imagine that's for you, Dr. Chen.
Yep. So that's a very good question. So unfortunately, buffer scales with how many grams of materials we produced. So we are looking at using inline concentrate. We're looking at buffer concentrate. We're also looking at making buffer inline. So both of the approaches. So far, we're working on both approaches. Yeah.
Thank you. Our next question is, would you consider the implementation of real-time release testing methods for your continuous biomonitoring platforms? And what type of tests/testing methods are you looking to explore in the field of on/inline monitoring and real-time testing? Again, I believe that's for you, Dr. Chen.
Yes, this is also a very good question. For biologics, the real-time release testing is a lot harder. So as I mentioned, for small molecules, tablets, there are already quite a few products approved by FDA. For biologics, because the testing is so complicated, it's very hard to do real-time. So right now we are doing, we have real-time capacitance probe, monitoring cell density. We have a real-time Raman, look at the metabolites, and on the process side, we look at the bioburden, we look at the product quality. But it's unlikely in the next couple of years, unlikely that we can do real-time release of biologics. So probably on release testing side, it probably still it's a combination of online and offline tools.
Thank you, Dr. Ribault. Do you have anything you'd like to add?
Yeah, a couple of points. Same technologies as the one mentioned by Chris Chen. NGS is a very interesting option as well to check on contamination in general, but also stability of the cell lines, and can be used in real time, very much like a PCR. Anyway, NGS, many people in the audience knows. At-line probes tend to be delivered more and more. Infrared measurements, Raman measurements, to look at what's going on in the bioreactor, metabolites, but not only metabolites. Checking on the increase in the molecule titer as well can be very interesting. But we're definitely moving towards the use of new technologies, at-line and in-line, and that fits very well into the scope of the continuous manufacturing, obviously.
Thank you. Our next question is, what is your next technology step moving forward? And I believe that would be for you, Dr. Revolt.
So next technology move, well, actually there's a couple. There's one on at-line probes, exactly what I just mentioned, actually. looking into what's going on in the bioreactor in real time. So it's one of our 3 next technology moves. The other one is definitely proceed with the intensified seed trained that I showed in the presentation today. The last one is not a manufacturing topic; it's a process development topic. It's a very general topic, so I won't be able to elaborate. But it's the implementation of new strategy in process development. So that process development timelines can be cut by about 25%, and it's a mix of new technology, automation, and data modeling as well.
Thank you. Our next question is, how do you address potential issues of extractables and leachables from disposable bioreactors? Dr. Chen, would you like to answer this?
Yeah, so WuXi, we actually screen for every cell line we select. That's part of the cell line selection criteria. So as I said, if you put this process forward, and then you wouldn't have any issue down the road with extractable leachables.
Yep, thank you. Our next question is, do you have experience producing clinical material with the continuous capture step, 3C PCC, if I understood correctly? That's our audience member. Dr. Chen, would you like to answer that again?
Yes, so we actually have already produced a few batches using continuous processing. We are able to achieve a 20 gram per liter for a very complex molecule. This is a bispecific antibody. We're able to achieve 20 gram per liter in a 17-day process, and it's a continuous capture with a off-the-shelf system. So this is essentially a 200-liter bioreactor is actually operating almost like a 4,000-liter bioreactor with this technology. We have successfully demonstrated that in the lab scale last year, and then now in the GMP scale this year.
Thank you. Dr. Ribault, would you like to add anything?
Yeah. So we are actually running a process that is continuous. It's a 33 days process, full perfusion bioreactor and continuous DSP. Well, contiguous DSP, meaning that some unit operations are still disconnected, but they run side by side. On the question that was, if I understand properly, on the 3 columns, we haven't run a project that I would qualify as clinical, if I understood well the question. I call that more a development mode. So yes, we've run not one, but processes using continuous capture, but it's more in a development mode than clinical mode. But clinical mode will follow very shortly because the technology is mature now. Our philosophy is really to make sure by running multiple projects and processes in a development mode that it's robust enough to go to clinic. We don't want to take a project and fail because the technology is not mature enough. So we usually take a year and can be up to 2 years, which is exactly what we did with the single-use bioreactors, to make a full evaluation with the number of model antibodies, hormones, cytokines, and so on, and make sure that we have no surprise when we run in routine.
Thank you. And our next question is, what is the scope of disposable downstream processing in the overall scheme of innovative biomanufacturing? Dr. Chen, would you like to take this first?
Yeah, so overall downstream, because of the scale and also because a lot of filtration and chromatography system in there, so it may not be that easy to do disposable unless you're truly going through continuous. Because with continuous, your column now is 10 centimeter or 20 centimeter, right? If you don't use continuous, your column is 1 meter. There's no way you can go disposable. So I think right now, if we implement continuous processing, then disposable downstream is actually maybe become feasible.
Thank you. And Dr. Ribault, do you have anything to add for this question?
Yeah, the question is interesting, and for me, it relates to many discussions we have where people tend to separate upstream from downstream. For me, the scope shouldn't be downstream or upstream. The scope should be manufacturing in general. If you don't have a good upstream, it doesn't make sense to have a good downstream. You need good upstream and downstream. So if it goes to continuous, there is a real benefit of having both continuous upstream and downstream, and a full alignment between what comes out of the bioreactor and what enters into the DSP process. So I would say that when we look at innovation, the scope is the scope of the process and not only of downstream. So we work in parallel on capture, on polishing, on TFF, on harvest, and obviously on the bioreactor as well. So I wouldn't exclude anything from the scope when we talk about innovation. Innovation is about innovating in all the process steps so that we have, in the end, a process handling that makes sense for the manufacturer. We don't want to be super fast on one step if we have a bottleneck right after.
And with that, we will actually conclude the question and answer session. But thank you, everyone, for your questions. Before I finish the webinar, I would like to ask our presenters, Chris and Sebastian, if they have any closing remarks. So, Dr. Chen, do you have any remarks for our audience?
It's a great event. Thank you.
And Dr. Ribault, do you have anything to say for our audience?
Yeah, thanks everyone for attending. I guess this is the first one of a long series of discussion. As you have heard through the two presentations, there are lots of things ongoing. Single use is only about 10 years old, more or less. We're at the very beginning of a lot of new innovation being deployed in the operations, in GMP activities. For me, it's the start of a very exciting journey.
Thank you. And I would like to take this opportunity to thank our presenters. Once again, that was Chris Chen, PhD, CEO at WuXi Biologics, and Dr. Sebastian Revolt, Senior Director of Global Delivery and Sales of BioReliance End-to-End Solutions at Merck, for sharing their knowledge with us. I would also like to remind our audience that you can view this and other webinars on demand by visiting biopharma-asia/on-demand-webinars. And if you are watching this on demand, then please feel free to send your questions over to me at stephen.edwards@biopharma-asia.com. I will get those answers sent back to you. Thank you, everyone. Have a great day.